This is a lecture on the anatomy of animal cells for Bio 101 for September 18th, 2020. And today we're going to talk about the cell theory, and then we're going to talk about the parts of the cell, starting with the plasma membrane, and then the functions of several organelles you can see listed here. So as we already discussed when we talked about the organization of living things, cells are the smallest living thing, and that is the first component of the cell theory. The second part of cell theory states that all organisms are made of one or more cells, right? And so if cells are the smallest living things, all living organisms are also made of cells. And then this is sort of the most important tenet of this cell theory. All cells have to come from a pre-existing cell. You can't make a cell out of nothing. And they have to come from a cell that already exists. And so cells being the smallest living things, all organisms being made up of one or more cells, and then all cells coming from a pre-existing cell are the three tenets of what's known as cell theory. And we're going to specifically be focusing on eukaryotic animal cells for this lecture. And eukaryotic cells, or cells that have a nucleus, are larger and more complex than prokaryotic cells. One, because they do have that nucleus to hold the DNA, but they also have membrane-bound organelles, which prokaryotic cells do not have. And so a eukaryotic cell is encased by a membrane, which you can see here in orange is the plasma membrane. But then inside of that membrane, the cell is filled with what's called cytoplasm, or the cytosol. And cytoplasm is a semi-fluid, it's kind of like a gel that fills up the cells. And this gel, the cytoplasm, is made up of about 70% water. And that is going to become important when we talk about how the membranes, both the plasma membrane and other membranes, are organized. And then within that cytoplasm, as I mentioned, in a eukaryotic cell, you'll find organelles, or sort of the organs or functional compartments of a cell. And the number and the types of organelles can vary between different types of cells. Some cells may have a lot of these mitochondria here. Some might have a lot more lysosomes. And usually the number and types of organelles are based on what the cell's function is, especially within a larger organism. And so our cells, like our neurons, actually have a ton of mitochondria because neurons need a lot of energy. And as we all talk about in a little bit, mitochondria are the sort of energy-producing organelle. And so depending on what your cell needs to do, it can have more or less of certain types of organelles. But it will have at least some of all of the things listed on the next or subsequent slides. So before we get into the inside of the cell and start talking about all those organelles, we're going to start at the outside, or the plasma membrane. And what the plasma membrane is a sheet of two lipids, which you can see here in like red and yellow. And that sheet of lipids has embedded proteins, which are in blue. And because this lipid sheet has two layers, it's considered a bilayer. And it's made of that specific type of lipid we discussed in last lecture, the phospholipids, which have phosphate groups facing outward and hydrophilic, water-loving heads outward, and hydrophobic or water-fearing tails inside, right? And so the way that this picture of the membrane is oriented is this is the outside of the cell here, and this would be the inside of the cell. And you can see this phospholipid bilayer, or this layer of lipids, that does have proteins embedded or basically inserted all the way through it. And those proteins can be seen in blue. And some of them span the membrane and have a hole in them to allow things to pass through that membrane. Some of them have little sugars hanging off the edge. Those are involved in helping your body recognize which cells are part of your body and which cells are foreign invaders. And the types and structures of proteins that are embedded in the plasma membrane can vary depending on the type of cell as well. And so the plasma membrane's main function is to just separate this outside environment from the inside environment. It's basically the thing that keeps the inside of the cell separate from the outside. And it does this by being what we call selectively permeable. We'll get to that in a second, right? Because this plasma membrane, we're going to go back to the structure, right? It's made of phospholipids. And phospholipids, to remind you again, have two different sides to them. They have a hydrophilic or a water-loving head region, which is seen in this blue circle. And they have a hydrophobic or water-fearing part, which are these tails, these gray sticks sticking out of this side. And whenever you put a phospholipids into water, which is what we just said, the cytoplasm is 70% water, cells are kind of bathed in water at all times. And when you place phospholipids into that water, it spontaneously forms the lipid bilayer that we see making up the plasma membrane. Because these hydrophilic heads will face the outside environment where there's water, and they'll face the inside of the cell or the cytoplasm where there's water. And those hydrophobic water-fearing tails will get buried into the core of the plasma membrane, making a kind of a two-layer situation with heads facing outward and tails facing inward. And since the function of the plasma membrane is to separate the outside world from the inside of the cell, it has to do that, but your cell can't exist in isolation. Some stuff does have to get in, and some stuff has to get out. We need to be able to move water and other nutrients into the cell so that the cell can make energy, right, and replicate. But we also need to remove waste products out of a cell, otherwise it will die. And so there's this idea or theory called selective permeability, where cells can specifically control what goes in and what goes out. And so phospholipid bilayers or plasma membranes are selectively permeable. They let some stuff through really easily, and other stuff they prevent from coming through. And so this is a selective control of what goes in and out of cells. Because if you let everything in, you would also be letting in some negative stuff, right? Toxins, big molecules, stuff that might actually harm your cells. And if you released everything from a cell, it might not be able to retain the nutrients it needs to survive. So there has to be some selectivity in the process of what goes into and comes out of a cell.